Hey everyone, welcome to Professor Long's Lectures in Anatomy and Physiology. Quick disclaimer, these videos are being shot very quickly, very crudely, in a very impromptu fashion with no editing because we're in the coronavirus shutdown. I've been saying this with every video. I know my students are tired of it, but these videos may end up on YouTube for a while, and anyone tuning in later, I don't want them to think this is the best that I can do. I just don't have time to edit, learn the software, and do any editing. So all of these videos are being shot from home because all of our all of our face-to-face classes have been switched to online, and the time frame is tight with all the other things that we have to do. I'm learning how to do testing software and a number of other things, and I'm dealing with students, emails and questions about exams and other things. So time is limited, so these are quick, they're impromptu, they're one takes. If you hear a dog bark in the background or a kid say, "Hey Dad, what's up?" it's because I'm shooting them at home. Bear with me, okay? We're gonna use these warts and all. Now, we left off in the nervous system on the bottom of page 70. We were talking about synaptic activity in the last video, and I have a worksheet up for my students, and there's a lot of information on page 70. You guys need to understand how synapses work, and once you understand one synapse, you understand them all. Just the name of the neurotransmitter changes, the name of the enzyme, and the name of the receptor. And then you need to know which ones are excitatory and inhibitory. We went over a very small sampling of neurotransmitters and synapses, but you can fill in the details later on. Now, at the bottom of page 70, we're talking about the glial cells, or what some people call neuroglia. And sometimes the word's pronounced neuroglia, tomato, tomato. So neuroglia also are abbreviated as glia. Glial cells are like the glue that holds the nervous system together. They do support for neurons. Remember, the nervous system is made up of two major classes of cells, neurons and neuroglia. We've been talking about neurons. Now, glia are somewhat, like I said, the glue that holds the nervous system together. They support the neurons in their functioning, physically, biochemically, nutritionally, emotionally. No, not emotionally, just kidding. There's emotional support glia. No, but they do a lot of biochemical, nutritional, and physical support. We're going to focus on 6 major glial cells, and really 5, but I'm going to mention one of them. So in the central nervous system, in our brain and spinal cord, we have 4 types of glial cells that are found there. One type of glial cell is called an astrocyte. Astrocytes get the idea, or get their name, from the fact that they are star-shaped. Okay, astrocytes do a lot of sort of physical support. They make up the framework that holds all the axons and the dendrites, almost like telephone poles supporting wires, similar to that concept. One of the things that a number of astrocytes can do is this. We've talked before, I believe we haven't. There's a part of your brain called the choroid plexus. The choroid plexus is a network of capillaries, very small blood vessels, that allow things to exit the bloodstream to nourish our neurons and things to come in to the bloodstream, like the neuronal waste and all of this other stuff. So we have to feed our neurons. We have to feed them all the nutrients, all the sugars and fats and proteins and amino acids. And water and beneficial ions flows out of our bloodstream and in between the cells. It forms the interstitial fluid of the brain. Those cells will consume all the nutrients, make waste, and dump their waste into the surrounding interstitial fluid. Well, if we don't wash it away, the waste would build up and poison our neurons. They would become septic in their own fluids. So one of the things we have to do is get stuff out of the bloodstream to feed our cells, and then from the cells back into the bloodstream so we can wash it away and clean it. This is how our nervous system and our circulatory system are going to kind of interact, is at the structure called the choroid plexus. So we have these specialized blood vessels that have a very thin wall, single squamous cell thick. They're capillaries. And all the nutrients and the good stuff will flow out of here. Ions, glucose, lipids, amino acids. Our neurons will take in all that nutrients to make energy, to rebuild, to make proteins, to heal, and they'll dump carbon dioxide. Oh, by the way, oxygen. They need oxygen for energy. They'll dump carbon dioxide and waste into this fluid, and it has to be reabsorbed into the bloodstream. Now, we talked early in part 1 about this concept called homeostasis, one of the central tenets of this course. And we talked about a range of conditions for homeostatic conditions. For example, we talked about temperature. There's a range of temperatures at which our cell's chemistry can function. That temperature can go up and down a little bit. 98.6 is the optimal temperature in Fahrenheit. And if it goes up a little bit to 99, we're okay. If it goes down a little bit to 98.8, we're okay. We can take small fluctuations. So there's a homeostatic range. If it gets too hot or too cold, that shuts down the chemistry of our cells and they die. That would be pathology. We also look at things like salt concentrations. Too much sodium and potassium can shut down neurons and neuron functioning because they use sodium and potassium and calcium and chloride. So ion concentration has a range here. We can look at oxygen concentrations. We can look at glucose concentrations. There's a range, okay? Now, some cells have a very wide range. They're very tough and hardy cells, like skin cells and other cells. You can mess with their conditions, and they're okay. Some cells have very narrow ranges of homeostatic conditions. Everything must be precisely perfect, or they start falling apart. They can't hang. I think of them as big wimps or weenies in the nervous system. The biggest wimps and weenies are neurons. You mess with their oxygen concentration, their sodium and potassium and glucose and lipid concentrations, and they don't function very well at all. Now, not only that, but if you think about the bloodstream, let's look at one blood vessel. Look at it like a river. And as water is flowing through this river, if I have people living down here, these would represent our cells, like neurons, and other cells in the human body. Eventually, this whole river circulates back around. And we have some filter organs, like the kidneys and our liver, which are going to clean that blood. But there are some cells that are dumping waste into the stream that other cells could be exposed to. Now, some cells are pretty hardy. They can take a little bit of it. But neurons can't. So neurons have to take this fluid, run it through a filtration membrane, filter it off, and then dump their waste back in. Okay? That fluid that gets filtered out of our blood is called cerebrospinal fluid, CSF. And the special blood vessels are called the choroid plexus. It means a network. So the choroid plexus is a special series of blood vessels that filter our blood, pull out the cerebrospinal fluid and leave all the rest in the bloodstream. And then the CSF will bathe our neurons in nutrients and ions and goodness, and then they dump their waste back into the bloodstream. So, there's a reason I mentioned the choroid plexus. Give me a second, my dog is barking. All right, I apologize, but again, I'm doing these at home and I'm not wasting 7 minutes of a video and starting over, or else I'd never get done. So now, when I look at these blood vessels of the choroid plexus, it would be like this little capillary. What helps form part of that filtration membrane that we filter cerebrospinal fluid out of our blood happens at these capillaries. So inside here would be our plasma, and out here would be cerebrospinal fluid. Well, it turns out that astrocytes are these little star-shaped cells, and they have these long extensions, or I should say short extensions, and they end up in these little feet-like structures that sit up against a capillary. And they can make several of these sometimes, not only on one capillary, but on others. And other astrocytes will form these little feet-like structures, and as we put them together from a number of astrocytes, they create this little filtration membrane. That membrane that allows us to keep large molecules from getting out into the cerebrospinal fluid, some trash and toxins and other things, but only let little tiny molecules through, into and out. They call that the blood-brain barrier. So we say that astrocytes help form the blood blood-brain barrier for CSF formation. Again, CSF stands for cerebrospinal fluid. It's written in the note set, so you can look at it. Okay. So astrocytes do some physical support of neurons, but they're also helping form the blood-brain barrier for the filtration of CSF. Now, a second type of glial cell that we can talk about is called an ependymal cell, and some people say ependymal cells. Okay. Tomato, tomato again. Ependymal cells can monitor CSF and they make adjustments to it. They have the ability, and they're found in certain parts of the ventricular system, but these ependymal cells they sort of line the third ventricle and some of the other chambers, the ventricular system and the central canal of the spinal cord, and it's like they constantly sample the cerebrospinal fluid and can make adjustments to the concentrations of certain things. That's a real generalized overview, but that's good enough for us for now. The third type of glial cell that's in the CNS that we can talk about is called microglia. Microglia are modified immune cells, or white blood cells. We usually abbreviate white blood cells as WBCs. And what they do is, just like any immune cell, anything that does sneak out through the blood-brain barrier into the brain that doesn't belong, they can gobble it up. They are phagocytic, and they cruise around the brain looking for trash and debris and things that don't belong, and they literally consume it, break it down, and digest it. They can also help digest old, worn-out, dying neurons. As we lose neurons, these guys can help digest them, just like any white blood cell can attack and digest any other cell. So the last of the four glial cells that we can talk about is going to be the oligodendrocytes. I usually do these third, but I wanted to do them fourth because it's going to lead us into another one. So let me erase some of this simply because I'm running out of room on the board. Pardon me, I dropped my eraser. And it's harder to write at the bottom. So now, you know the function of astrocytes. They help form the blood-brain barrier for the formation of CSF, and they do some physical support of neurons. Ependymal cells do some biochemical or nutritional support by adjusting CSF. And microglia protect the neurons by cruising around and acting like immune cells. Now, the last of the four CNS glial cells that we can talk about are called oligodendrocytes. Now, before I talk about that, let me mention in the peripheral nervous system, in the PNS, we have two major glial cells that we're going to talk about. One of them is called Schwann cells. Well, you've already heard about Schwann cells. They myelinate axons in the peripheral nervous system. So if I have a neuron, soma here, that's going to taper down at the axon hillock, and then the axon goes and branches into telodendria, and we have a synapse down here, these cells actually wrap around and cover parts of the axon with a fatty substance called myelin. They make up the myelin sheath. I talked about this before when we talked about action potential velocity and saltatory conduction. These would be Schwann cells. They're filled with a fatty substance called myelin that insulates the neuron. And so the action potential as it travels here would jump to this node of Ranvier, jump to this node of Ranvier, and they assist action potential velocity. Now there's a couple of other minor things that scientists have discovered that they can help with. But ultimately, Schwann cells myelinate axons in the peripheral nervous system to aid in action potential velocity. Well, in the CNS, we have myelination. But there are no Schwann cells found in the central nervous system. It's oligodendrocytes. They myelinate axons in the CNS. And therefore, they aid action potential conduction in the central nervous system. And one of the major differences is in the CNS. If I were to look at, let's say I have an axon here traveling through the board towards you, and I have several axons like this. One oligodendrocyte, I'll put a little D for his name, can actually wrap a myelin sheath or wrap its membrane around several or a few, hence the term oligo, axons. So in the PNS, a Schwann cell myelinates a single axon. In the CNS, this oligodendrocyte can myelinate a few, hence the term oligo. They have a few branches and they myelinate axons in the CNS. Damage to the myelin sheath would affect how action potentials travel here, and that's one of the things that happens in multiple sclerosis, is that in MS, a lot of the neurons will lose their myelin. The Schwann cells seem to unwrap, so to speak. It messes up for a while, and then they'll rewrap, and then they'll unwrap and rewrap. And over that period of time, every time, it's kind of like if you tried to unwrap a Christmas present to look at it, and then you go back and you try to rewrap it, you're never going to make the wrapping look like it did originally. And every time you do it, it gets worse and worse. Over time, it gets progressively worse. It's one of those episodic diseases that happens in short episodes or sometimes long episodes. But it happens in an episode and then it disappears for a while. And then you'll have another episode, it disappears, another episode. So it's an episodic disorder, but each episode gets a little worse than the previous one. And it's tough. I have some friends that deal with it. Anyway, so you can understand how myelination affects neurons in the CNS. So now, the last of the neurons, or the neuroglia that we're going to talk about, are called satellite cells. Satellite cells, I tend to think of them as similar to ependymal cells in that they do some nutritional or biochemical support and adjustment for the cells in the peripheral nervous system. Okay? All right, so that should cover the glial cells that we've talked about. I'm going to stop here, and we're going to do the very last video in the section of the nervous system. So I hope you learned something. I hope this was helpful, and I hope to see you on the flip side. So thanks for watching.